QIMR Berghofer acknowledges the traditional and cultural custodians of the lands, waters and seas across Queensland, and we pay our respects to elders past and present. We support the cultural knowledge, determination and commitment of First Nations communities in caring for health and well-being for millennia.
Welcome to QIMR Berghofer's 80th anniversary podcast. I'm Claire Blake, and we're celebrating 8 decades of bold ideas, real science, and the brilliant men and women who made it happen. We did start in a leaky old army hut, and now we're tackling cancer, decoding the brain, tracking infectious diseases, and rewriting what's possible in medicine. We'll dive into the true stories, funny, messy, inspiring, from the people who made it all happen, and those carrying the torch into the future, because science isn't just labs and data, it's human. And it's happening right here. Today, Professor Greg Anderson. He is a world leader in iron research, longtime head of our Iron Metabolism Lab and former deputy director, and he has some stories, nearly 3 decades worth, in liver disease and the lessons learned through mentorship and collaboration. Welcome, Greg.
Thank you, Claire.
How did an undergrad from Newcastle end up fascinated with iron biology and join QIMR?
Well, like many things, you sort of fall into it. I was an undergrad in Newcastle, did my honours, the last year of my undergraduate degree, and I was applying for various scholarships. I wanted to go overseas and study. So I applied for various things, including a Rhodes Scholarship. I got to the short list, but the guy who won it the year I was there was some guy who went on to nothing. I think his name was Tony Abbott.
Never heard of him.
No, he's not known very much these days. And I applied for some other scholarships and ended up going to Canada. But while I was waiting for scholarships to come through, I needed to earn some money, so I took a job at QIMR. I was only here 6 months. I disappeared for some time. But one of the things I did during that 6 months, in fact, the first day I worked here, is I met the woman who was later to become my wife and has been for now almost 41 years. So that was one bonus from that job. After that term overseas finished, of course, there was imperative to come back to Brisbane. And I took a job then working with Laurie Powell and June Halliday, then in the Department of Medicine at the University of Queensland.
Ah, so there's the connection.
And then later, when Laurie became director of the Institute and his group moved to the Institute, that's when I moved with them. So that's when I started my long-term association with the Institute.
You started at 81. By 95, you had your own lab. Where did the fascination with iron come in? Or was it just the mentorship?
Probably more the latter, because just about any area gets interesting when you look into it in enough detail. After I had worked with Laurie and June for some time, I enrolled in a PhD and did my PhD. I was interested in iron. I continued working in iron, and then I went overseas postdoc and worked in iron there in quite a different area, genetics of yeast and iron metabolism. Not long after I returned to Australia, a year or two after that, June Halliday retired, and then myself and others, particularly Grant Ram, started to run our own laboratory groups.
Your contribution working with Grant Ram was hemochromatosis and the danger to the liver.
Hemochromatosis was for a long time not considered a real disease entity. It was thought to be a variant of alcoholic liver disease. Laurie Powell, starting in the 1960s, didn't believe that was the case, and ultimately, of course, he was vindicated. Laurie's group, of which Grant Ram and myself were an important part over the last 40 years or so, has contributed to understanding hemochromatosis in many ways, and that's including looking at the basic mechanisms leading to iron loading in patients with hemochromatosis. And I tended to go down that path, looking at dietary iron absorption and why we take up too much iron from our diet. The other end of that is when you've got too much iron, it causes damage to the liver and other organs. And Grant really embraced that, and his career followed that path. So we still both continued to work in hemochromatosis and work together throughout our time at QIMR together, but we looked at different aspects of it.
And great friends as well.
Yes, we've been friends for a long, long time.
Was there any antics that we can share here?
Lots of antics, share? No. We used to do a lot of things behind June Halliday's back that if she knew about it, she never told us about it. But we had a lot of fun.
Information was much more slowly coming in those days. Was that a challenge when you look at it from today's lens?
Hindsight always changes things, of course, but at the time that was what you did. That was state of the art, but we didn't have the easy access to journals that we used to have. QIMR had an extensive library, which has disappeared now because it's not needed anymore. You can get everything you want online. Things were a lot slower. If you would send a manuscript off for publication, it's a process that could take 6 months to a year, and these days it can be really quite quick. So you work with the technology that you have available at the time, and that's just the world we lived in then. You can look back in retrospect and say it was primitive at the time, but at the time it was state of the art.
You're one of the few that has unique perspective because your wife actually worked at the original Army surplus hut over on Vic Park.
I don't know too much about exactly what happened in that building, but the Institute was very small at those times. I joined the Institute when we moved into the second building, and you might say the first permanent building on what's now the RBWH campus. And the Institute was a different place then. It was before the time that the science really embraced the molecular era that was brought to the Institute by Chev Kidson when he became director. And there were a lot more what you might call traditional things going on. For example, the Institute now has a mosquito control unit. In those days, we had mosquito taxonomists as well, like Pat Marks, for example. There was also a mite taxonomist, so he was looking at mites and the viruses they carried. So there were a lot of those more traditional taxonomy technologies as well. When Chev came, those people were ultimately moved to museum roles, and the molecular aspects of the work took over.
That was a massive change.
Yes, it was, and big change in technology as well. The Institute used to have a goose, for example. That was used to provide blood because the goose blood cells were used in virus agglutination tests as part of virus diagnostics. So bleeding of the goose was a regular activity, and the goose was kept in the animal house. But the goose has long since gone or been eaten, perhaps, I don't know.
Did the goose have a name?
I never was on such intimate terms with the goose that I learned its name.
I'm going to find photos of the goose.
Good luck.
There were other things. Grant Ram has mentioned at Christmas they had the Olympics across the road and they took the load out of the centre of the centrifuge and used it as a shot put.
Yeah, that came along later, but there was some creative use of laboratory or excess laboratory equipment, older laboratory equipment for the games, yes.
There's some things in place to make sure that doesn't happen any longer, which is a bit disappointing, I guess, isn't it?
Health and safety, you know.
And just looking back from where you are now, that you happen to work with people like June Halliday and Laurie Powell, you must be so grateful that they're the connections you made, or maybe they feel that way about you.
Yeah, they were great people to work with. June was a pioneering scientist, well, pioneer for women in science. One of the first, perhaps even the first Fulbright scholar from Australia, female Fulbright scholar from Australia. So she was very accomplished and always a very strong advocate for women in science. She didn't suffer fools gladly. Outsiders perhaps considered her a bit of a tyrant, but she wasn't really like that at all. She just had standards and expected people to adhere to those standards. You know, if you did right by her, she was incredibly supportive of you and would really foster your career.
Like any good leader.
Yeah. And Laurie the same. But I suppose June was the one on a day-to-day basis. At least initially I had more to do with because she was leading the lab-based part of the research and Laurie was off doing the clinical side of the work.
They're extraordinary triumphs over your career, like showing that iron removal helps with liver disease and advanced cases. Now looking back, it seems extraordinary.
Iron removal aspect has been going on a very long time and it's a very simple procedure. So, in the old days, people used to use leeches to treat diseases. Well, leeches would work very well for hemochromatosis because they can take out a lot of blood. Despite that phlebotomy, the removal of blood, remains the mainstay of treatment for hemochromatosis because it's very simple, has few risks, and it's very cheap. If you're someone who's diagnosed with hemochromatosis and have maybe 20 or 30 grams of iron in your liver, which is a lot because you would normally only have maybe 3 or 4. I'm sorry, a hemochromatosis person might only have 3 or 4. It can take a long time to get rid of that iron because you need to take a lot of blood out to deplete your iron stores. There are other ways of looking at that, and there are some novel molecular approaches and drug approaches that have been developed, but they're still relatively expensive. There will be more treatments coming along that may improve a lot of patients, but still at the moment, blood removal is the mainstay of treatment.
I know that David Fraser, who now runs the Iron Lab, is very passionate about finding other better ways, and in both sides, low iron and high iron as well. It must be good to have somebody to really take on your work, because I know lots of people leave and there's nobody that's following their passion.
Now, it certainly is, and you can never guarantee that someone is going to follow on the types of things you've been doing. David and I have worked together for a very long time now. He was a PhD student of mine and postdoc with me, and now has his own group, as you say. He's become particularly interested in the other side of things, having too little iron deficiency, which of course is an enormous problem worldwide with billions of people who are iron deficient, and also looking at iron metabolism and iron handling in pregnancy and early postnatal life, which are clearly critical times for development. So he's pursuing some really important areas.
And we love David, of course. He's very passionate and an incredible communicator of his work in lay terms, which is not something every researcher has the ability to do.
The way I usually say that is he talks a lot.
He won't mind you saying that, I'm sure. Is it my imagination, or do iron researchers have a robust sense of humour?
I think it's good to have a sense of humour, and sometimes it's important to keep things light. You need to have fun at what you're doing. If you don't have fun in doing it, you may be in the wrong career.
Lots of experiments don't go as planned. Do you remember something extraordinary that went wrong?
I don't think we ever had any spectacular failures. I think for most researchers, that shouldn't be the case if you've carefully considered what you're going to do and you've planned it appropriately. And I remember someone telling me once that he had postdoced with a Nobel Prize winner, and the thing that struck him most when he went to that lab is that they seemed to spend very little time doing experiments, but a lot of time in the pub. And what they were doing in the pub is talking through the experiments, looking at all the angles, etc. So by the time they got to the lab to do the experiment, they looked at all the possibilities of outcomes beforehand, so that experiments were very targeted. Hopefully, if you've planned well, you don't have spectacular failures. When I first came to QIMR, I was working with a virologist called Peter Walker, who was then a junior virologist, went on to become a senior virologist. And we were working on RNA fingerprinting of dengue virus. So that meant we were looking at the molecular profiles of different strains of dengue virus. And you would take some RNA out of the virus, you would cut it up with an enzyme, separate the fragments on a gel, usually a gel-like support, and you'd apply an electric current there to separate them. And these gels ran for a long time. And one day we set up one of these gels, and on that day there was a wine tasting in the city, and Peter said, look, we really should go to this wine tasting while this gel's running, which we did. We went off to the wine tasting. We tasted quite a lot of wine, and at the end Peter said, look, I'll go back to the lab and take the gel off, and you can go home. And when I got into the lab in the morning, before Peter, the gel was smashed into hundreds of pieces on the floor. And, of course, this was a radioactive gel as well. Again, some of the safety standards were not as high then as they are now. Peter came in, but he said, okay, we just do it again. That's probably the most dramatic failure I've come across in my work.
Talking to lots of other researchers, it really becomes clear that extraordinary collaboration that you have at lunch, in between experiments, or over the road, or weekend picnics has been incredibly fruitful to progressing the science. And we don't really have that as much now. We're much bigger and it's harder to do.
When I first came to QIMR, a number of the younger scientists of the Institute were involved in what was then called the Moreton Island Protection Committee. Moreton Island is now largely a national park, but in those days it wasn't. And there were regular trips several times a year over to Moreton Island to camp out and enjoy Moreton Island. So a lot of the younger people used to go on those as well.
Oh, that'd be fabulous. You've had lots of students, lots of successes. Did they teach you things?
I think the most important thing with students is to make sure they don't believe what you tell them. And I mean that in the sense as they shouldn't just trust everything you say. They need to go and look at the evidence themselves and make up their own mind. And sometimes when they do that, they come back with a different perspective that you may not have thought about. That enables you to design different experiments and look at different things. So it's beneficial to you that they establish their own independent thinking.
What about your mentors?
There's a lot of people who have influenced me over the years. In Newcastle, Tim Roberts was a young lecturer there who is still around, emeritus. I've been in contact with him in recent years, still very supportive. Of course, June and Laurie, the core of my scientific career, which took me from a new PhD student to someone who was known internationally in the field. They were always strongly supportive, but also allowed independence. They didn't prescribe exactly what you wanted to do. They encourage you to think creatively and follow your own interests.
You were part of shaping cancer, infectious and chronic disorders programs. Was it easier then than it is now? There's so much more red tape.
So the basic unit of work or group, if you like, of the Institute is the lab. I think it always has been. And at various times, we have had different units or departments or divisions or programs. They keep getting changed. The fact that every few years they get changed, perhaps an indication that there's no perfect way to do this. Challenges change over time. I think the programs at the moment seem to be working very well. In the earlier programs that we were running, they were useful to get people who were at least working in a broadly similar area or using similar methodologies, etc., together. But they tended to be largely administrative units with a scientific overlay, just a more efficient means of getting information out to the different labs. But the lab itself remained the core entity of the functioning of the Institute.
You're now an emeritus professor with QIMR Berghofer. Your days in the lab aren't what they used to be. You still have a space. Do you miss those days of being in the lab?
I suppose it's a long time since I have done experiments with my own hands, and probably the people stand back in fear if they see me put on a lab coat. It's different. You do miss it. QIMR is a very vibrant scientific community, and sometimes I worked in places with a much smaller cohort of scientists, and there's not quite the same vibe as there is in a large and dynamic institute. But I have plenty of other interests that have kept me occupied, so I'm very pleased to retain an association with the Institute, and we still have some work ongoing in collaboration with David. There also always comes a time where you need to stand aside a bit and let the next generation come through, and I'm happy to do that as well.
I know we could do a whole nother podcast or three on spiders. That's your other passion, something you really, really love, and you work with the museum as well, don't you?
I have appointments at the Queensland and Western Australian museums to work on spider taxonomy, so that's a lot of fun, and I have a lot of time in the field, ranging from the desert up to the tropical rainforest. So that is a very different kind of science than the sort of science I've been doing for the last 4 decades or more, but still very enjoyable.
What's your favourite spider?
It's like, who's your favourite child?
Oh.
I can't tell that. My favourite spider at the moment is one that does not yet have a name. It has a very unusual method of catching its prey, and is probably, as far as we're aware, the fastest acting snare of any animal known.
It's Australian?
It's Australian, but similar spiders are found throughout Southeast and East Asia as well.
Spiders are just so clever.
Well, they have a lot of pre-programmed behaviors. There are some spiders that can learn and learn very well, but most of them are just following what nature has programmed them to do.
Well, we just don't want the funnel web learning any new tricks.
No, they're a pretty basic sort of spider.
Oh, really? Who's the most clever in the world of spiders?
There is a jumping spider called the genus Portia, And the species we have in Australia is called Portia fimbriata. It has a very plastic neural mechanism. It can learn new behaviors. It has very good memory, or remember things that it did at least 6 months earlier. So it's amazing for something that just has this tiny ganglion in its head can do what it can do. And it can learn new behaviors, and that's been shown experimentally.
Well, forget how to do the jumping, Spider.
I won't forget how to do the jumping, but its prey capture is very diverse and it can learn new ways of catching its food.
We're glad to still have you and your passion and your long legacy and all your triumphs as a part of QIMR Berghofer. Is there anyone that, past or present, that you'd love to just sit down and listen to a little bit of their research?
Lots of people over the years that have influenced me in different ways. Of those I've never have been able to talk to or listen to, people like Galileo or Marie Curie would be terrific. I've always been a great fan, though, of Alfred Russel Wallace. So Alfred Russel Wallace should be a lot more better known than he is. He is considered the co-discoverer of evolution by natural selection with Charles Darwin, but Darwin gets all the credit. Wallace was slightly junior to Darwin, but he was an incredibly creative thinker. He had enormous hardship in his long life. He lived until he was 90. Probably at the time he died in 1913, he was considered the most famous scientist in Britain. He could have been buried next to Darwin in Westminster Abbey, but he said he didn't want that. He wanted to be buried in his local cemetery. His scientific friends had a plaque in his honour erected next to Darwin's grave in Westminster Abbey, so you can see that there. Buried in Broadstone Cemetery, just outside of Bournemouth. And I have dragged my wife along to Wallace's grave to pay homage. So mounted above his grave is a petrified tree trunk, or part of it, over 2 metres high, that was found on the southern English coast. No one quite knows why that ended up there. Probably the family thought that would be something of interest to him. The unfortunate, although Wallace might find this amusing things, is that this tree trunk looks enormously like a giant penis. And I think he'd probably have a bit of a chuckle to himself if he actually saw what it looked like. Wallace did an enormous number of interesting things, and I'd really love to have lunch with him.
Happy 80th anniversary, Greg, and thanks for joining us today.
And happy 80th to QIMR Berghofer.